Alrighty, so let's begin here with the first set of checkpoint questions, and we'll go over the answers. So describe the barriers for reabsorption and secretion. Be sure to include all plasma membranes that must be crossed by the substance being reabsorbed or secreted. So first off, we want to establish that the barriers are the exact same. The only difference is the direction of movement, okay? And so the layers that we talked about, the membranes that had to be crossed, was the epithelial cell, so the plasma membrane on the apical surface of that epithelial cell. Remember, let's jump back here. Remember, the apical surface is actually facing the lumen. So we got to go through the apical surface, we got to go through the cell itself, the basolateral surface, the peritubular space, and then the endothelial membrane of the capillary, right? So those are the three layers that we're going to cross for reabsorption and secretion, but just in opposite directions. The second question here, oh no, I think I skipped. Oops. Where did they go? Okay. The second question was, describe the general mechanism for water reabsorption. So in terms of reabsorbing water, we said that sodium was actively pumped across the basolateral surface by the sodium-potassium pump. Chloride followed passively, and then because of the movement of those two solutes, then water followed according to osmosis passively because of that gradient. So that is how water is reabsorbed. And then finally, define the terms transport maximum and renal threshold. So we said transport maximum was the rate of movement. Remember, this is a rate, whereas renal threshold is a plasma concentration. So you want to kind of get that, differentiate those two differences in the terms here. So transport maximum is the rate of movement when all the transporters, all the carriers are fully saturated. Whereas renal threshold is the plasma concentration of substance X. We talked a lot about glucose as an example, when we start to see spillage of this substance because the transporters are saturated, right? So transport maximum is the rate of movement at which maximum saturation happens, and then renal threshold is the plasma concentration at which that saturation happens. Okay, let's go to the second set. Okay, so name the three structural differences between epithelial cells of the proximal and distal tubule, the structure of which tubule is more favorable for exchange. So let's go back to this slide briefly. So we said there were three kind of main differences between the proximal tubule and the distal tubule. One was the nature of the tight junctions. So in the proximal tubule, we have leaky tight junctions. In the distal tubule, we have tight junctions. And that is indicating to us that the proximal tubule is unregulated, the distal tubule is regulated or controlled. Another structural difference is the brush border. So the brush border, which is formed by the microvilli, is only present in the proximal tubule and not in the distal tubule, another indicator of an adaptation for increased reabsorption, right? This is increasing the surface area. And then the third difference would be the shape of the cell. So I talked briefly about these being taller columnar cells. Columnar cells are more suited or adapted for reabsorption as opposed to the shorter cuboidal cells of the distal tubule. So the shape of the cells is different, the apical modification of the cells is different, and the nature of the tight junctions are different. And all of those differences point to the proximal tubule being more favorable for reabsorption. Alrighty. Okay, I think that was the second part of the question. The structure of which tubule is more favorable for exchange? That would be the proximal tubule. Question 2, the clearance of which two substances can be used to estimate the GFR? The clearance of which substance can be used to estimate renal plasma flow? Now, we did not speak about renal plasma flow, so you can kind of eliminate the second part of this question. But in case you are curious, it's going to be PAH, para-immunohepatic acid. So the substance that I mentioned that renal specialists put into the body, inject into the veins, and they use that to measure how much blood is flowing to the kidneys. That's the renal plasma flow. Okay, we did not speak about that, but just in the event that you're interested. The second part of the question, what two substances were used to measure GFR? Now, inulin is a great measurement for GFR because it's the exact same clearance rate as our GFR. So inulin is one, and then creatinine is another substance that renal specialists often use to measure GFR, simply because it's greater than GFR, and it's going to tell us if GFR is less than it should be. So inulin and creatinine are two substances where the clearance rate is used as an indicator or a measurement for GFR. Okay, question 3. If the clearance of molecule X is greater than GFR, was it reabsorbed or secreted in the tubules? So we said that if the clearance was greater than GFR, right, then the clearance, if you think about what clearance means, then this substance is showing up more in the urine, right, at a greater rate in the urine than it's being filtered. So that, if you just think about that simply, it's coming from somewhere else. So it must be secreted into the tubules. Whereas if it's being cleared at a rate that's less than what it's being filtered, then it must be going somewhere, right? And so we understand that it's definitely being reabsorbed if the clearance is less than the GFR, whereas if the clearance is greater than GFR, it's certainly being secreted, okay? And we said if the clearance is 0, it's being reabsorbed 100%. If the clearance is the same as GFR, it's neither reabsorbed nor secreted. And that's why inulin is such a great estimator of GFR, because inulin is neither secreted or reabsorbed. And so it's going to give us a very good estimate of what our filtration rate looks like, of what our GFR is. The fourth question, describe the innervation of the bladder, internal urethral sphincter, and external urethral sphincter. Voluntary inhibition of micturition through innervation to which of these structures? So innervation of the bladder itself, the detrusor muscle, is through the parasympathetic branch of the nervous system, the autonomic nervous system. Innervation to the internal urethral sphincter is also under the autonomic nervous system, but the sympathetic branch of the autonomic nervous system. And then innervation to the external urethral sphincter is via the somatic nervous system, which is under our voluntary control. And so the second part of the question, voluntary inhibition of micturition through innervation to which of these structures? And so it's basically asking us which structure is being innervated during the guarding reflex. What is that? That guarding reflex that we talked about, where this is what's preventing you from going to the bathroom on yourself, right? So that is innervation to the bladder, to the detrusor muscle, which is being inhibited. So in order to guard the bladder, we inhibit the parasympathetic innervation to it, whereas when we're ready to void the bladder, we're going to inhibit the internal urethral sphincter, inhibit the external urethral sphincter, but stimulate the parasympathetic to the bladder. Okay, hope that makes sense. All right, so let me see if there are any questions, and then we'll jump to the third set of questions. So the third set of questions were these. Okay, so question 1, where does most water reabsorption occur in the renal tubules? Where does the regulation of water reabsorption occur in the renal tubules? So the first part, straight away, we know it's the proximal tubule, about 70% of the water that is reabsorbed through the entire nephron, 70% of that happens in the proximal tubule. And we talked about the mechanism, and we talked about the adaptations of those cells for reabsorption. Where does the regulation of water reabsorption in the renal tubules occur? So where does the regulated water reabsorption happen? That's going to be the distal tubule and the collecting duct. Alrighty. We know that water is also reabsorbed in the descending limb of the loop of Henle, that is unregulated, and so it's going to be a less of a percentage, but it's going to be unregulated. Question 2, compare the movements of solute and water across the epithelial cells lining the descending limb of the loop of Henle and those lining the ascending limb of the loop of Henle. Explain how this differential movement allows establishment of an osmotic gradient in the renal tube, in the renal medulla. So we talked about the establishment of the medullary osmotic gradient, and this is just the gradient of the osmolarity of the tubular fluid from the cortex, where the proximal tubule and distal tubule are, to the medulla, where the very tip of the loop of Henle is. And the establishment of this gradient happens because of the differing permeabilities. Remember, the descending limb of the loop of Henle is only permeable to water and not permeable to solutes, whereas the ascending limb, the limb that curves back up, is only permeable to solutes and not permeable to water. So in order to establish this gradient, the ascending limb pumps solutes into the medullary interstitial fluid. The descending limb will then try to correct that gradient by pumping an equal amount of water into the interstitial fluid, into the medullary interstitial space, and then more water moves into the tubule, and that happens again and again and again until we get to this steady state. And at that steady state, the osmolarity gradually increases until it's the highest at the tip of the loop of Henle, and then it gradually decreases until it's lower. This is the whole point of the medullary interstitial, of the medullary osmotic gradient. Let me go back to this slide. Oh, where is it? This slide. So we can see here that the whole point of this gradient is so that now at the end of the loop of Henle, the osmolarity of the tubular fluid is less than 300, which is less than the osmolarity of plasma. And remember, we said in order for water to be reabsorbed, there must be an osmotic gradient between plasma, where that water is going, and the tubular fluid, where that water is coming from, right? So now we have a solute gradient, we have an osmolarity difference or an osmolarity gradient so that water can move. Alrighty. And then the final question here, can pure water be excreted as urine? Explain. So the short answer is no. Pure water can never be excreted as urine because we looked at the range of the osmolarity of urine, right? We said in its most dilute form, with zero reabsorption taking place, the osmolarity of urine is 100 milliosmoles, meaning that although it's quite watery, there is still 100 milliosmoles of solutes dissolved in this solution, in this filtrate here. The highest osmolarity in its most concentrated form, the osmolarity is about 1400 milliosmoles. So although we have that range of osmolarity, we can never excrete pure water as urine. And that just goes against the fact that urine is eliminating waste products, right? So there must be solutes and waste products, things that are secreted present in that urine, and so it can never be